Hi everybody, welcome to another Chats with Children. I hope you're all safe and well. Today I'm delighted to have another catch up with Steve Burton, who's the CEO of Astrea Bioseparations, and we're going to be talking about some of the changes within the business over the last it's 4 months now, I think, actually, Steve, isn't it? So first of all, Steve, lovely to meet you again. How are you?
I'm very well, thanks you, Rizwan. It's great to catch up with you again.
Yeah, so last time you and I caught up was in June of this year, and you had just been acquired by Gamma Biosciences. I know a lot's been lots changed in the business since then, but before we start talking about that, perhaps you could give people who weren't able to catch the first interview or don't know much about Astrea a bit of a background of the company and your role within it, if that's okay.
Yeah, sure. So the company, as you say, was acquired by Gamma Biosciences last November. It owes its origins back over 30 years ago to a point where it was created as originally as a spin-out company from one of the Cambridge University departments. And for 20 years of the company's life, we were a part of Chromatic Life Sciences, now Liminal Biosciences, where we had a focus of developing purification products, principally affinity adsorbents, and also supporting the Chromatic company with process development expertise. We were very much involved in the development of the plasma protein fractionation program that Chromatic had, and as a part of that, I think we developed something in the region of 15 new chromatography adsorbents for that program of work. But at the beginning of last year, for a variety of reasons, it became clear that the time has come to move on again, and that we were reached the point where we were a viable business in our own right, and so that the company was put up for sale. And fortunately, just at the time that Gamma Biosciences was formed, so Astrea was, as we're now called, formerly Chromatic Bioseparations, now Astrea Bioseparations. But we were the first acquisition that Gamma Biosciences made. And the way that we've been working together with Gamma in the recent months, we're evolving into the downstream processing part of Gamma Bio. So the recent acquisitions that have been made within Astrea Bioseparations, because they're both in the downstream processing space.
Fantastic. Well, let's talk about those acquisitions, because I know you've had a couple of acquisitions since we last spoke. So talk me through those acquisitions and the reasoning behind them, if you like.
Yeah, sure. So the first one was ELS and Essential Life Solutions. And this was a company founded by Dan Yukon in the US. And Dan developed a very well-respected range of chromatography columns, the SNAP columns, and these are quite widely used, for both our research and development applications and also for small-scale process applications as well. And we have been working with Dan for quite a number of years now, so we were very, very aware of him and his business. And he has also been marketing the Evolve column range that Astrea has in the US. So it seemed to make sense that we bring Dan into Astrea. So we completed that acquisition in August, towards the end of August this year. So the SNAP column range, obviously still with ELS, but now as a part of the Astrea Bioseparations group of companies. And we're now in the process of promoting those, not just in North America, but globally now through the Astrea network. And of course, Dan has his offices, his facilities, laboratories, just outside of Boston in Stoughton in Massachusetts. So that's a very convenient location to have a North American base. So as
Sure.
a consequence of this acquisition, Astrea now has offices in just outside of Boston in the US. So very, very pleased to be working with Dan. He's very experienced, very knowledgeable, and a great asset to the
Yeah,
company.
he's a great guy. Anyway, I've interviewed him, and actually, if I did an interview with him, as you know, and if people want to watch that interview, there will be a link to that interview as well as part of this video post as well. So we'll look out for that. Now, you've also had a second acquisition though, because you've just acquired. And now I might pronounce this incorrectly, so forgive me if I'm happy for you to correct me. Nanopareil, is that right? Or
Correct. Yes.
Yes, right.
Nanopareil. Yes.
Nanopareil. Right. So tell me more about Nanopareil and the products and technologies and why you've brought those into the fold.
Yeah. So Nanopareil is a very interesting company and has very interesting technology. We became aware of them not long after we were acquired by Gamma Biosciences ourselves. And we have historically had an involvement with non-woven fiber materials as a substrate for the attachment of things like ligands, for example, as an alternative to chromatography beads. And we were aware that the advantages of these non-woven fibers are a combination of the fact that you have that there's no pores as such, not like in a chromatography bead.
Right.
So there are through channels, basically the spaces between the fibers. And the fibers themselves are non-porous. So the binding is to the outer surface of the fiber. So the smaller the fibers, the greater the surface area to volume ratio. And nanofibers have an enormous surface area.
Right.
We could say more in a moment, some of the advantages and benefits that offers. But this is very unique technology. There's very, very few companies globally that have any involvement with these nanofiber materials for bioprocessing.
Right.
So as soon as we became aware of this, we thought, right, we have to meet with this company. This is a perfect fit with Astrea. So they're based in South Dakota. Unfortunately, we managed to meet with them just before the lockdown. We were unaware of what was coming down the road at that point.
Yeah.
But we met up with them and just just hit it off right from the first meeting. We had a very similar outlook and similar view of things, and
Right.
And realized that we could work together. So we went through the process of due diligence, and we acquired Nanopareil. That was announced just a couple of weeks ago at the end of September. So again, very, very pleased to have secured that the ownership of Nanopareil. They have really fantastic technology, and I'd be happy to say more about that.
Fantastic. So does that mean that, because obviously Astrea has always been known as a chromatography for its chromatography products, the resins and the columns. So does this mean that you're expanding more into filtration technology?
That's a good question. At some point, maybe yes, but not right now.
Right.
The reason why we've acquired Nanopareil for their nanofiber membrane is primarily as a next generation substrate, sort of base material for our chromatography, our ligands and surface chemistries. So we're using it as a surface platform to attach our chemistries onto, with a view to performing adsorptive chromatography. So, but instead of using beads, we're now using derivatized non-woven fibers. So that's the main focus for the business, but we are aware that these nanofiber materials do potentially have applications as more conventional filtration materials, but that's not for now. Our focus right now is on the use of these as an improvement over conventional chromatography beads.
Right. Well, you mentioned a little bit earlier about some of the advantages, but what are the main advantages of nanofiber membranes, and also how will they benefit manufacturers of biological products?
Yeah, well, there's actually several benefits to these materials, and they really are game changing in terms of the impact that they're going to have on bioprocessing. So, as I've mentioned, the main features of these materials are that the membranes themselves, and this is like a non-woven fibrous mat that's fashioned into a membrane. The fibers themselves are nanofibers, so they're incredibly small fibers. By themselves, they're so light they would just float in the air. They would be unusable as at the point where they're produced in any kind of filter. But Nanopareil have spent a number of years and very cleverly developed a way of fashioning these incredibly light, fine nanofibers into what is actually quite a robust membrane sheet,
Right.
Without any sort of degrading the performance of the fibers in any way. They're still nanofibers. They're not sort of glued together to form one large fiber, for example. And so they have this huge surface area. And what that means is that binding is virtually instantaneous.
Right.
So as soon as an analyte, such as it might be a protein or a viral particle or something like that, as soon as it comes into contact, it binds straight away. It doesn't have to diffuse into pores to bind. So that means that the binding is very rapid, which means that in terms of what's called residence time, so when one is flowing liquid through a packed device, for example,
Right.
the residence time for a chromatography column is normally measured in minutes. So usually it will be somewhere between 3 and 5 minutes, a typical residence time for a chromatography column. With these nanofiber membranes, the residence time is measured in seconds. So that the flow rates that these will support are extremely high. So that means you can process very large volumes of material with a very small sort of size unit. They can support extremely high flow rates and still have very effective binding. And then another advantage is the fact that this large surface area means that when it's derivatized with ligands, for example, and ion exchange groups, it has a very high capacity. So with ion exchangers, for example, we see more than double the capacity of a conventional ion exchange chromatography column. So you have this interesting combination of very high flow rates, very rapid binding, and very high capacities as well. And then to add to that, the membrane itself obviously has to be porous in order for liquid to flow through.
Sure.
But it's not the fibers themselves that are porous. It's the spacing between the fibers that creates the pores. And by adjusting that spacing, it's possible to enable the transmission of relatively large biomolecules. So now not just proteins, but macromolecular assemblies, viral particles, vaccines, things of that nature. So they're incredibly useful materials. What we can do with these, based upon the properties, is really, is completely novel, and we're extremely excited by the potential of this material.
Right. And how does the Nanopareil fit with your existing technology and product portfolio then? So, obviously you've got these now. How are they going to fit with your portfolio?
It fits actually very well, and as I've just described, the main intention here, the focus here, is the derivatization of these materials with affinity ligands, and we, of course, Astrea, as for many years now, has established a very well used platform for discovering, developing affinity ligands. So that's something we've been doing now for over 30 years. We have many examples of successfully implementing adsorbents at scale. So we don't just have the ligand technology, we have the ability to scale up ligand manufacture, to scale up the immobilization and produce these things in industrial quantities, essentially. So we can apply all of that knowledge and know-how and the affinity ligands to these nanofiber materials now. So there's a very nice fit there. So going forward, we will have ready-to-use devices
Yep.
for both chromatography adsorbents and nanofiber membranes, and we will be focusing these on the applications to which they're best suited to. So there isn't going to be a case of one size fits all here. There will be a preference of either a bead or a membrane, depending upon the application. But we're in a position to provide both of these in pre-packed, ready-to-use formats.
Fantastic. And when would you expect to launch the first nanofiber products, and what surface chemistries will be offered?
Yeah, so the first surface chemistries that we're looking at are almost certainly going to be the ion exchanges, both anion and cation. They're available, we have quite advanced prototype materials right now that Nanopareil have developed. And they're of general utility. Of course, ion exchanges are used in many aspects of bioprocessing. And particularly for the cell and gene therapies as well. So I think that the focus initially is going to be on the ion exchanges, but we'll be following up then with more specific affinity adsorbents. In terms of timescale, these are fairly well developed at this stage, but we still have some further work to do on this. So we're targeting probably the first half of next year for the initial launch of the first product, but the first range will be following on from that. But there's already been over 6 years of development work performed on these, so they're already at quite an advanced stage.
Great. And so one final question. Things always happen in threes normally. So is there anything else down the pipeline that you're looking at the moment? I mean, obviously you won't necessarily talk about.
Well, yes, there are some things we're looking at right now to further expand. I can't go into details right now, but I will say watch this space.
Oh, well, fantastic. Well, look, as always, thank you very much, Steve, for taking the time out to talk to me. I appreciate it. I know you're very, very busy, and to fit this in is very, very difficult, but I appreciate taking time out. I hope everybody found that useful. It was really interesting. I'd never heard of nanofiber products before, so I learned something new today as well. So I kept thinking when you're talking about glue actually and spreading it out over a large surface and how it binds very quickly. In my head, that's what I could visualize when you were talking about it. It's quite interesting. So thank you very much. I hope everybody found that really interesting. Steve, as always, thank you very much for your time. Look after yourself. Hopefully we'll catch up again soon.
Great, and great to talk to you again, Rizwan. It's always a pleasure.
It was a pleasure for me as well, Steve. And I hope you all found that useful. If you've got any questions for Steve, please, as always, leave comments below the video. There will be a link to the previous interviews, as I said, with Dan and to Steve from our initial conversation way back in June. Time has changed so much since then. And also to the Astrea website as well. So you can find out more about the solutions and technologies that Steve was talking about. So it'll all be above the video as links as well. So as always, thank you for your time. Thank you for watching, and stay well and stay safe. Bye-bye.
Bye.